Assalamualaikum and greetings to fellow lecturers and friends. Welcome to my final year project video presentation. My name is Nurul Syairah binti Nur Syamrizal Baharin, a final year student of Bachelor Science Honour Biology. The title of my final year project is Antibacterial and Antifungal Effects of Calendula officinalis and Eucalyptus globulus essential oil. I am supervised by Ms. Nur Tabitha binti Sheikh Nasir. This presentation will highlight 5 chapters that consist of an introduction, literature review, methodology, result and discussion, and lastly, conclusion and recommendation. Let's begin with Chapter 1, Introduction. Calendula officinalis and Eucalyptus globulus are a plant that has various therapeutic properties. Calendula plant parts such as leaves and flowers has therapeutic properties such as antibacterial and antifungal. Meanwhile, Eucalyptus globulus plant parts such as leaf extract and essential oil has proven to contain antibacterial and antifungal properties. Furthermore, gram-negative and gram-positive bacteria such as E. coli and Bacillus are responsible for causing food contamination such as food spoilage, as well as the fungus Aspergillus can affect food directly and indirectly, especially moist food. So this can be dangerous to one's health. Even though synthetic pesticides have a high rate of effectiveness, but the health and environmental effect of synthetic pesticides become an issue in this study. Synthetic pesticides not only have toxic effect on non-targeting organisms such as beneficial bacteria, but in a long-term period of use, their effectiveness decreases against crop plants. Therefore, it is critical to restrict the misuse and use of synthetic pesticides to protect biological diversity. These issues have forced the creation of alternatives to synthetic pesticides using natural products. Botanical pesticides have become a choice to replace synthetic pesticides because they have secondary metabolites such as alkaloid, steroid, terpenes, and etc. They also possess antibacterial and antifungal properties that can be used to treat bacteria and fungal contamination on plant crops. And not to forget, they degrade faster than chemical pesticides and have a low probability of killing beneficial organisms, which make them more environmentally friendly. The significance of the study is this study exposed the public to value that Calendula officinalis and Eucalyptus globulus hold and can produce a lot more natural products. Moreover, this will provide added value for the plants, such as helping the farmers grow healthier crops while reducing losses due to disease caused by bacteria and fungi. The objective of this study is to determine the antibacterial activity of Calendula officinalis and Eucalyptus globulus essential oil on gram-negative bacteria which as E. coli and gram-positive bacteria such as Bacillus. It is also to determine the antifungal activity of Calendula officinalis and Eucalyptus globulus essential oil against the fungus Aspergillus. Chapter 2 is literature review that covers several aspects. According to Ashulayan et al. 2018, pot marigold is the common name for the plant Calendula officinalis, which belongs to Asteraceae family. According to Yasin et al. 2021, a short-lived aromatic herb, Calendula officinalis, grows up to 80 cm tall and has waxy, smooth stems. The plants are hermaphrodites with tubular flowers that are typically darker orange, 5 to 70 cm long and hairy on both sides. Next, the component of Calendula officinalis. According to Pandey et al. 2017, terpenoids, a large group of secondary compounds, are crucial in creating a barrier against pathogens. Next, according to Shaw et al. 2020, the tall evergreen tree species known as the blue gum tree is called the Eucalyptus globulus. It is a medicinal herb that has a fragrant smell. The important component of Eucalyptus globulus. According to Shaw et al. 2020, because it is effective against a variety of microorganisms and has broad-spectrum antibacterial properties, eucalyptol is a substance that is crucial to the Eucalyptus globulus plant. Move on to methodology. First, dilution of essential oil. Calendula officinalis and Eucalyptus globulus were diluted using coconut oil and a dilution chart, and made into 20%, 40%, and 60%. Second, prepare nutrient broth. This is used to cultivate E. coli and Bacillus and assess the antibacterial activity of Calendula officinalis and Eucalyptus globulus. Third, prepare agar media. Agar media for fungi and bacteria are different. Potato dextrose agar is used to make microbial media for fungi, while Mueller-Heaton agar is used to make microbial media for bacteria. Fourth, mud filling standard. For bacteria, The bacteria were diluted with normal saline to achieve the 0.5 microgram standard. Meanwhile, 0.001% twin was used for fungi and achieved 1 microgram standard. Fifth, both bacteria and fungi were checked for their turbidity using spectrophotometers. 6. Bacteria and fungi culturation. 100 microliters were taken up using a pipette and spread into the agar media to grow bacteria and fungi lawn. These steps were repeated with sterilization. 7. Dish diffusion. The dish will be saturated with 25 microliters with 3 different percentages, 20%, 40% and 60% of Calendula officinalis and Eucalyptus globulus essential oil, and sited in the inoculated microbial media. After that, the petri dish will be incubated at 37 degrees Celsius for 24 hours for bacteria strain and at 25 degrees Celsius for 72 hours for fungal strains. By measuring the growth inhibition zone diameter in mm using a ruler for the tested species, the microbial inhibitory effect will be recorded and compared to the antibiotic standard. Ampicillin disc and gentamicin disc were used as a positive control for antimicrobial and antifungal to control isolated microorganisms. Coconut oil will be considered as negative control. 8. Minimum inhibitory concentration MIC was determined for both essential oil. All tests were performed in triplicate. The MIC value will be determined and expressed in percentage, which is 40% for Calendula officinalis and 60% for Eucalyptus globulus. Lastly, statistical analysis. The data obtained were analyzed using SPSS software. The data from bacteria and fungi was run using two-way ANOVA. Next, we jump into the result of this study from the table shown with the use of Calendula officinalis essential oil. The highest mean inhibition is 11.5 plus minus 0.5, which is against E. coli using 40% concentration, and the lowest mean inhibition is 10.83 plus minus 0.76 using a 20% concentration of essential oil. Furthermore, the activity of Calendula officinalis against bacillus shows no inhibition zone, 6.0 plus minus 0.0 using all three concentrations, 20%, 40% and 60%. The mean inhibition of ampicillin as positive control is higher against E. coli, 8.83 plus minus 0.29, meanwhile 7.0 plus minus 1.73 against bacillus. This shows ampicillin is more susceptible towards E. coli. The higher mean inhibition of negative control is 10.0 plus minus 0.0, which is against bacillus. But with the use of Calendula officinalis alone towards bacillus, there is no antibacterial activity appeared. This shows that the negative control used has antibacterial properties and a stronger antibacterial activity than Calendula officinalis. Next, using Eucalyptus globulus essential oil. The highest mean inhibition 11.33 plus minus 0.58 using 60% concentration, and the lowest mean inhibition shown by the E. coli towards Eucalyptus globulus essential oil is 10.33 plus minus 0.58 using 40% concentration. The Eucalyptus globulus essential oil also work on bacteria Bacillus subtilis with the highest mean inhibition of 10.33 plus minus 1.15 using 60% concentration. Meanwhile, the lowest mean inhibition is 8.83 plus minus 0.29 using 20% concentration of essential oil. By comparing a 20% concentration of Eucalyptus globulus against Bacillus subtilis 8.83 plus minus 0.29 and against E. coli 10.50 plus minus 0.8, E. coli shows highest mean of inhibition, which shows that Eucalyptus globulus is more susceptible towards E. coli compared to Bacillus subtilis. The largest mean of inhibition shown by ampicillin is 11.0 plus minus 2.65, which proves that ampicillin has good antibacterial properties against bacillus compared to E. coli. But the antibacterial properties of ampicillin are not as strong as Eucalyptus globulus against E. coli. In this case, the negative control also shows good antibacterial activity against both bacteria, but higher for E. coli, 10.67 plus minus 1.15, and bacillus 10.0 plus minus 0.0. According to the statistical analysis, the relationship between sample and bacteria, sample and concentration, bacteria and concentration, and bacteria sample and concentration shows a significant difference with a p-value less than 0.05. From the result of multiple comparisons of different concentrations, there are no statistically significant difference between concentrations of 20%, 40%, 60% ampicillin and negative control with p-value more than 0.05. Stated that the R-squared value 0.786 shows a positive value. This means that the value of one variable increase is associated with an increase in the other variable. On aspergillus, using Calendula officinalis essential oil, the highest mean of inhibition in this study is 9.33 plus minus 2.31 with the use of a 40% concentration, and the lowest mean inhibition is 6.33 plus minus 0.58 at a 60% concentration. With the use of Eucalyptus globulus essential against fungi aspergillus, the highest mean inhibition is from 60% concentration with 19.33 plus minus 11.55, and the lowest mean of inhibition is 12.13 plus minus 5.32 using 20% concentration of Eucalyptus globulus essential oil. Calendula officinalis and Eucalyptus globulus essential oil shows no reactions toward positive control as well as negative control. This proves that Calendula officinalis and Eucalyptus globulus essential oil has high potential antifungal properties compared to gentamicin, which shows no inhibition zones against aspergillus. According to statistical analysis, the inhibition zone of both sample, Calendula officinalis and Eucalyptus globulus, shows a significant difference in antifungal properties between both essential oil with p-value 0.032. In this study, Calendula officinalis shows an antibacterial activity against E. coli but not Bacillus. According to Ashlayan et al. 2018, it has been proven that Calendula officinalis extract has antibacterial properties where it inhibits the growth of E. coli. Although Calendula officinalis contains bioactive compounds such as flavonoids, terpenoids, and essential oil, the specific antimicrobial mechanism and spectrum of activity may not be effective against Bacillus strains. Bacillus species, including Bacillus cereus and Bacillus subtilis, have unique characteristic and resistant mechanisms that may render them less susceptible to the antimicrobial compounds present in the Calendula officinalis. Calendula officinalis has a strong antibacterial properties compared to the commercial antibacteria. The p-value between ampicillin and 40% concentration of Calendula officinalis essential oil, which has the highest mean of inhibition, is 0.07. This shows that there are no significant difference between those two antibacteria that indicate that Calendula officinalis essential oil at 40% concentration is more effective than ampicillin towards E. coli. Eucalyptus globulus shows antibacterial activity against both bacteria, E. coli and Bacillus. According to Uitm et al. 2020, Eucalyptus globulus essential oil inhibited the growth of S. aureus, B. cereus, and E. coli. The p-value between ampicillin and 60% concentration of Eucalyptus globulus essential oil, which has the highest mean inhibition, is 0.122. So there is no significant difference between those two, which also means that 60% concentration of Eucalyptus globulus essential oil is more effective than ampicillin towards E. coli. This study also shows inhibition by negative control. The researchers found that coconut oil exhibited significant antibacterial effects, inhibiting growth and reducing its viability. According to Anzaku et al. 2017, it is because coconut oil might be attributed to its high content of lauric acid, a medium-chain fatty acid known for its antimicrobial properties. On the other hand, Aspergillus, Calendula officinalis, and Eucalyptus globulus essential oil shows an antifungal activity against Aspergillus. This can be proven from the previous study. According to Ibani et al. 2018, Eucalyptus essential oil reacts with Aspergillus at a minimum inhibitory concentration of 4.575 mg per ml. From the figure shown, we can see gentamicin that acts as positive control does not show any inhibition zone against Aspergillus. The p-value obtained is 0.156, so there is no difference between gentamicin and 60% concentration of Eucalyptus globulus essential oil that has the highest mean inhibition, and shows that 60% concentration of Eucalyptus globulus essential oil is more effective than gentamicin towards Aspergillus. As for Calendula officinalis essential oil, the p-value obtained is 0.025. There is significant difference between gentamicin and 40% concentration of Eucalyptus globulus essential oil that has the highest mean inhibition against Aspergillus. This may be due to the stability of antibiotics, the duration and temperature of incubation, and resistant mechanism found in the tested bacteria. As a conclusion, the essential oils of Calendula officinalis and Eucalyptus globulus have good antibacterial and antifungal properties with their effectiveness against both gram-positive and gram-negative bacteria as well as against fungi as has been discussed previously. This study has provided ample evidence of the antibacterial and antifungal properties of Calendula officinalis and Eucalyptus globulus, indicating their potential as important resources for the creation of antimicrobial therapies. It is recommended for future study to consider a more in-depth understanding of Calendula officinalis and Eucalyptus globulus essential oil, such as their mechanism of action, as well as investigating specific components present in them and their interaction with bacteria and fungal. And combination therapy may provide a clearer insight into their antimicrobial effects.